Hello everyone. Till now we have discussed about the structure of plasma membrane, about its lipid components, protein components and also some of the important features of plasma membrane. Today we are going to discuss about membrane transport. That means we will discuss in detail how the transport of the molecules takes place across the plasma membrane. So we have already seen that plasma membrane is selectively permeable. That means it allows the passage of or the movement of the selective molecules through it. And this selective permeability maintains the internal composition of the cell. Plasma membrane is actually a barrier and it does not allow the free passage of all the molecules between the cell and its external environment. We already know that plasma membrane has two important components, lipid components and protein components. So first we will focus on this phospholipid bilayer. This phospholipid bilayer allows the diffusion or it allows the passage of hydrophobic molecules. Also, the passage of small uncharged polar molecules. It is slightly permeable to large uncharged polar molecules. But it is impermeable to ions. So, phospholipid bilayers allows the passage of these molecules, hydrophobic molecules, small uncharged polar molecules and large uncharged polar molecules, but does not allow the passage of ions. We have seen that plasma membrane allows the passage of ions. So, the question arises how it takes place. This is because in the plasma membrane, transport proteins are also present, either in the form of channel proteins or carrier proteins. And these transport proteins allow the passage of these ions across the plasma membrane. Characteristics of lipid phase diffusion and protein mediated transport are different. Transport across plasma membrane is of two types: passive transport and active transport. Passive transport occurs along concentration gradient. So in passive transport, metabolic energy, or we can say ATP, is not required. In case of active transport, it occurs against concentration gradient. So here metabolic energy is required. Passive transport is further classified into two types: simple diffusion and facilitated diffusion. Simple diffusion. In simple diffusion, molecules dissolve in the phospholipid bilayer and then diffuses across it. So, it does not involve any membrane protein or transport proteins. It occurs along the concentration gradient. We can determine the direction of the simple diffusion by the relative concentration of any molecule inside and outside the cell. This is cell. Inside, the concentration of any molecule is C1 and outside it is C2. If C1 is greater than C2, then the diffusion occurs from cell to outside. If C2 is greater than C1, then the diffusion occurs from outside to cell. And if C1 equal to C2, then the diffusion does not occur. It is non-selective process. Any molecule that dissolves in phospholipid bilayer diffuses through it. Rate of simple diffusion is given by Fick's law. J is equals to minus D delta C by delta X. J is equals flux per unit area. D is equals diffusion coefficient. It is expressed in centimeters square per second. Delta C is concentration difference and delta X is thickness of membrane in case of membrane transport, or it may be distance. Molecules that diffuse across plasma membrane by simple diffusion are gases like O2 and CO2, hydrophobic molecules like benzene, and small polar and uncharged molecule such as water. Most of the molecules that are present in the cell are either too large or too polar to diffuse through the membrane by the simple diffusion. So their movement across the plasma membrane requires assistance of certain membrane proteins. These membrane proteins are known as transport protein. These transport proteins can be channel proteins or carrier proteins. And this type of the diffusion is known as facilitated diffusion because it is facilitated by these membrane proteins, or it takes place with the help of these membrane proteins known as transport proteins. Facilitated diffusion also occurs along concentration gradient. It is specific process because the transport protein does not allow the movement of all the molecules. The rate of facilitated diffusion is more than simple diffusion. Let us compare simple diffusion and facilitated diffusion. Simple diffusion occurs along concentration gradient. Facilitated diffusion also occurs along concentration gradient. It is passive because energy is not used. Facilitated diffusion is also passive. In simple diffusion, no transport proteins are involved. In facilitated diffusion, transport proteins are involved. Simple diffusion is non-specific. Any molecule that is soluble in lipid bilayer passes through the plasma membrane. Facilitated diffusion is specific because it involves transport proteins and molecules which can pass through these transport proteins diffuses across the plasma membrane. Rate of simple diffusion is less. In facilitated diffusion, rate is more compared to simple diffusion. When we plot a graph between the rate of the transport and concentration difference across the membrane, in simple diffusion we get straight line. This indicates that as the concentration of the solute increases, concentration difference increases, and this increase in the concentration difference increases the rate of the simple diffusion. When we draw a graph between the rate of transport and concentration difference in the facilitated diffusion, we get parabola. The rate reaches to the maximum and then further increase in the solute concentration does not increases the rate of facilitated diffusion. So in simple diffusion, no saturation is achieved. As the concentration of the solute increases, the rate increases. But in case of facilitated diffusion, saturation is achieved. This is because this facilitated diffusion involves transport proteins, and in the plasma membrane, these transport proteins are limited in number. As the concentration of the solute increases, the rate increases, but when all the transport proteins are occupied, further increase in the solute concentration does not affect the rate of facilitated diffusion. Types of transport protein for facilitated diffusion or transport proteins that mediate facilitated diffusion. One is carrier protein. Also known as transporters or permeases. These transporters non-covalently binds to specific molecule on one side of membrane. Then transporters undergo conformational change. This allows the passage of molecule through the membrane, and then that molecule is released on the other side of membrane. It can be better understood from this figure. This is plasma membrane, transport protein is present, specific molecule is present outside the cell, it comes and binds with the transport protein. Then the transport protein undergoes conformational change. This allows the passage of this molecule across the membrane, then it is released inside the cell and the transport protein again gains its normal conformation. So carrier proteins are stereo specific. Some molecule shows stereoisomerism. These are present in D form and L form. Binding site of the carrier protein will bind to one of the isomeric form but will not bind to the other isomeric form. This is known as stereospecificity. One of the carrier protein is glucose transporter. This is present in various isoforms. One form is the GLUT4 that is present in the skeletal muscles and adipose tissue. This glucose transporter is not present in the plasma membrane, but it is present in the intracellular vesicle. When the level of glucose increases in the blood, then insulin hormone is secreted. This insulin hormone then stimulates it. It fuses with the plasma membrane, and now the GLUT4 is present in the plasma membrane and it increases the uptake of sugar. Second type of the transport protein that mediate the facilitated diffusion is channel protein. These form open pores through the plasma membrane and allows the free diffusion of any molecule of appropriate size and charge. Now the comparison of carrier protein and channel protein mediated transport. In case of the carrier protein mediated transport, rate of diffusion is less. In case of channel protein, rate of diffusion is comparatively more. It is more stereospecific. Channel protein is less stereospecific. Carrier protein is saturable. Channel protein is non-saturable. Depending upon the type of molecules transported by the channel proteins, they are of different types. One is ion channels. These specifically transport inorganic ions. These ion channels are not permanently open. Some are voltage gated ion channels. These open in response to change in electric potential. Ligand gated ion channels. These open in response to binding of ligands. In many plasma membrane of animals, water channels are present, which helps in the osmotic flow of water. And these water channels are known as aquaporins. Ionophores. These are small, hydrophobic, lipid soluble molecules that dissolve in the phospholipid bilayer and increases its permeability. These are mostly antibiotics that are produced by the bacteria to kill target organism. These increases the permeability of the phospholipid bilayer and make it so permeable or so leaky to the ions that it becomes difficult for the target organism to maintain the gradient of some ions like Ha+, Na+, K+, and other ions that is essential for the lives, and hence it is difficult for the organism to survive. One of the example of this antibiotic is valinomycin. These ionophores are of two types. One is mobile ion carriers. These are hydrophobic molecules with hydrophilic cavity, and these shield the ions and helps it transport across the phospholipid bilayer. Second is channel formers. These dissolve in the phospholipid bilayer and thus forms the hydrophilic channels and helps in the passage or the transport of the ion across the phospholipid bilayer. Active transport. Active transport occurs against concentration gradient. Of solute molecules. It is mediated by carrier proteins. Since the molecules are transported against the concentration gradient, so metabolic energy is required. It results in the accumulation of solute molecules on one side of membrane. Carrier protein mediated facilitated diffusion is different from active transport. Let us see their comparison. Facilitated diffusion is selective. Active transport is also selective. Facilitated diffusion is passive. Active transport is active process. It occurs along concentration gradient. It occurs against concentration gradient. It is saturable. Active transport is also saturable. It involves transport protein. In active transport also, transport protein is involved. In facilitated diffusion, entropy increases. In active transport, entropy decreases. Active transport performs three major functions in the cells and organelles. First is the uptake of energy-rich molecules and other essential nutrients from outside, even though the concentration is low outside compared to the inside. Helps in removal of waste materials, secretory products, and sodium ion from inside to outside the cell, even though the concentration is more in the outside environment compared to the inside of the cell. Helps in maintaining constant optimal concentration of some inorganic ions such as hydrogen ion, potassium ion and calcium ion in the cell. In this way, active transport helps in maintaining internal composition of cell. There are two types of active transport: primary or direct active transport, secondary or indirect active transport. First is primary active transport. Here the transport is directly coupled to metabolic source of energy such as ATP. Example is sodium potassium pump that is present in plasma membrane. It involves sodium potassium ATPase, which hydrolyzes ATP. This is the plasma membrane sodium potassium ATPase non-cytosolic region, and this is the cytosolic region. Here each molecule of the ATP hydrolyze is coupled with the outward movement of 3 sodium ion, that is movement of 3 sodium ion from cytosolic to non-cytosolic region, and inward movement of 2 potassium ion. from non-cytosolic to cytosolic region. In this way, the intracellular concentration of sodium and potassium ion is maintained. Secondary active transport. In secondary active transport, uphill transport of one solute is coupled to the downhill flow of other solute. Uphill means against the concentration gradient. When one solute moves against the concentration gradient, then energy is required. So it is endogonic. And it is coupled with the downhill flow of the other solute. So it is exogonic. Then this solute is actively pumped by the primary transport. We can understand the secondary active transport by this figure. Here, the concentration of H plus is more in the non-cytosolic region compared to the cytosolic region, and concentration of this solute S is less in the non-cytosolic region and more in the cytosolic region. So this solute S is moving uphill, that is against the concentration gradient, and its movement is coupled with the movement of H plus downhill. Then this H plus is actively pumped from the cytosolic region to the non-cytosolic region to maintain high concentration of H plus in the non-cytosolic region. And the pumping is active, so hydrolysis of ATP takes place. So it is primary active transport because here direct source of energy, that is ATP, is used, and here it is secondary active transport because here energy that is released by during the downhill movement of H plus is used in the uphill movement of solute S. So here indirect source of the energy is used. So it is secondary transport. Example is the sodium glucose transporter. In this, sodium moves downhill and glucose moves uphill. Then this sodium is pumped against the concentration gradient from the cytosolic to the non-cytosolic region by the sodium-potassium pump. Depending upon the movement of the solutes, it may be symport, that is when the two solute moves in the same direction like here, and antiport if they move in the opposite direction. So this is all for today. In the next video, we will discuss about vesicular transport. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.